In early 2018, Regeneron, along with collaborators AbbVie, Alnylam, AstraZeneca, Biogen, Pfizer, and Takeda, formed a major research consortium to fund the generation of genetic exome sequence data from the 500,000 volunteer participants who make up the UK Biobank health resource. Each collaborator has committed $10 million to enable a dramatic acceleration of sequencing timelines, with Regeneron covering the balance and conducting the sequencing effort. The sequencing data will be paired with detailed, de-identified health and medical records from the UK Biobank resource. And it includes enhanced measures such as brain, heart, and body imaging to create an unparalleled resource for linking human genetic variations to human biology and disease. I'm not done yet, just a couple more paragraphs, bear with me. This unique approach and collaboration amongst industry dramatically speeds up the drug development process by enabling faster genetic target identification and validation. Recent advances in sequencing technology and integration of rich data resources mean there are infinite discoveries to be made that could potentially impact human health. Representatives from the group of collaborators join us today to discuss the thought process behind the group's novel approach to building its data resource, how they intend to open up the data for wider access and publication, and the importance of ethically managed genetic sequencing projects for medical progress. I'd like to welcome to the stage our moderator, Carrie Reinertsen, Vice President of Strategic Alliances at Regeneron, and her panel members. Michael Norson, Head of Business Operations and Strategic Planning at Regeneron Genetics Center. Paul Nioy, Senior Director of Translational Research at Alnylam. Slave Petrosky, Vice President and Global Head of Geome Analytics at AstraZeneca's Center for Genomics Research. Morten Sugard, Vice President of Target Sciences and Technologies, Worldwide Research and Development at Pfizer. And Joseph Stukowski, Executive Director of Translational Bioinformatics at Bristol Myers Squibb. Please welcome our panel.
Thank you. So Carrie Reinertsen, as she said, Head of Strategic Alliances at Regeneron. And it's great to be here today and tell you about an effort that our companies have established and what we think is really a unique relationship amongst our groups and the UK Biobank, and an activity that is really not like it in the industry in pharmaceutical and academic relationships. Maybe as a first step, I'll turn it to you, Mike, and you can talk a little bit about the history, describe the consortium, and what we're doing together.
Yeah, sure. So as Carrie said, thank you for having us. We're excited to be here to talk about the consortium that we formed in early 2018. Specifically, what the group up here has done is we've used funding from over 8 pharmaceutical companies throughout the world to accelerate the genetic sequencing of the UK Biobank under a pre-competitive consortium model. For those who aren't familiar with the UK Biobank, it's an effort in the UK, obviously, that's backed by the Wellcome Trust Fund that over the past decade or so has collected 500,000 genetic samples of DNA samples from volunteers throughout the UK, and has also paired it with electronic health record, really detailed data. And I think the other thing that's really unique about the UK Biobank is they've invested heavily in building imaging data across the body, specifically the heart, the brain images that are really unique. And being able to put all these things together just creates this unparalleled resource that is really available throughout the world. So we looked at that, so that was a very exciting opportunity if we could find a way to partner to sequence that data. So each of the companies here, as we approached each company, it's a massive project to do on our own, and if we're able to band together and each company could contribute, we could leverage the sequencing technology that Regeneron has built in-house to accelerate not only the sequencing of this data, but actually doing the project itself. So that was really essentially how the thought of the project came together. In terms of one of the other things that's really important here for a company's perspective of wanting to invest in this is working with the UK Biobank. There is a brief period of exclusivity on that data, and then once that period of exclusivity is over, we'll work with the UK Biobank to make that data available for all research to apply through their application process. We've also committed also to any sort of significant publications, or excuse me, significant insights or results that come out of the research. We'll make those available for the larger scientific community as well. And so far it's been a great partnership. As of this fall, we've sequenced over 50,000 of those DNA samples. And with the help of the group here, we're going to be able to finish all of the samples in terms of all 500,000 sequenced by the end of next year, which is just something that we couldn't have done on our own, and I don't know if any of you guys could have done it on your own as well. So we're really happy to have partnered together on this effort.
Yeah. And maybe I'll just give a chance for each of you to talk a little bit about the activities within your companies, how you've been thinking about big data and genetics, and how this fit within each of your efforts when we kind of first got together and talked about the consortium. Paul, if you want to start.
Yeah, so I can start us off. So, Alnylam, I think there's sort of really two aspects of this that we found really, really compelling. The first was that we're a company that aims to develop treatments for genetic disease. In fact, the programs that we have that are furthest along are all very much focused on helping individuals that carry certain genetic changes that influences the risk of developing disease. And so what was really exciting about UK Biobank was that it was a chance for us to sort of look in a population like that and just understand, well, if you have this genetic change in your genome, what happens to you? If you look in the medical records, how are these people diagnosed? And we suspect actually in many cases that individuals are underdiagnosed. So one of the really compelling things is, can we help to change that by understanding what these individuals are diagnosed with and sort of raising awareness of how these diseases present, helping physicians do a better job of actually diagnosing patients. So that's one aspect. The other is we're obviously very excited to try to discover new targets. So understanding human disease in more detail through the lens of genetics, and then using that knowledge to say, this is the target that we should go after with our next series of drugs. So it was those two things that I think we were really excited about, and it's great to be part of this effort.
Hi, Slava Petrovsky from AstraZeneca. So I agree with everything that Paul said, but just to give you a bit of insight of AstraZeneca's genomics initiative, where this falls under. So within AstraZeneca, genomics is at the heart of our drug discovery programs. In 2016, we established a genomics initiative which had the bold ambitions to analyse the sequence data of up to 2 million individuals, of which up to 500,000 are actually participants of our own clinical trials, AstraZeneca and Medimmune. We realised very early on that to achieve that bold ambition would require close collaboration with academic and industry partners who are experts in the field. And that's where this LIFE consortium opportunity really stood out as a great opportunity for AstraZeneca. It provides us with the clinical data sets to allow us to accelerate, as was mentioned before, new genetic target identification, to validate targets already in our programs, and also to understand how we can embed precision medicine, which is this concept of, as Paul alluded to, matching patients to the medicines that are most likely to benefit them based on the molecular diagnosis that they've been identified through genomics or other omics.
Okay, so Morten Sokkat, Pfizer, so they lead a group called Target Sciences and Technologies there, and kind of as we think about human genetics, there's studies that have been done, both by us and others, that show that if you work on drug targets that have strong human genetics and biology underpinning, you have a dramatically better success rate when you get into the clinic, particularly phase 2, which is the biggest issue for us, but also in general for the industry. We're not as smart as we think we are quite often. Of course, we try and do our best for diseases like rare disease and oncology with single molecular drivers. There sometimes it's relatively straightforward, particularly for a common disease. It's really challenging sometimes to understand where do we actually try and make a new medicine. So what this kind of resource helps us learn is what are the drug targets we could be working on, what are disease progression biomarkers, and of course at the end of the day, as several colleagues touch upon, with precision medicine, I mean, we hope to be developing better medicines for patients, and also being better at identifying the patients who can benefit from our medicines. End of the day, of course, that's why we are here. And I think this is UK Biobank is probably the leading resource in the world, for large population-based resource. And so we're really excited. I think we'll talk more about that in detail.
So my name is Joe Sostakowski. I'm with Bristol Myers Squibb. I've been there for about 3.5 years now. And during that time, BMS has made a significant investment in genomics and genetics as it relates to oncology, which is a large part of our portfolio at BMS. And in just those 3.5 years, we've seen sort of our understanding of tumor genetics really change the way that we think about classifying and treating different cancers, the way that we think about precision medicine in terms of getting the right drugs to the right patients at the right time. And we've recognized that there's an opportunity for similar resources and investments to have impact on our non-oncology portfolio, so the work that we do in diseases like cardiovascular and fibrosis and immunoscience. And we've been looking around for some time for a resource like this. So when our colleagues at Regeneron reached out to us and described the unique nature of this data that they were planning on generating, not only in terms of its scale, but in terms of the depth and breadth of clinical and phenotypic information available through UK Biobank, we jumped at the chance. In terms of the way that we're thinking about using the data, it's very much in the way that my colleagues have just described. How do we better understand the molecular mechanism of disease? How do we translate that into novel targets that are more likely to be effective when we drug against them? How do we understand patient response to therapies so that we can make sure that we're treating patients with the drugs that they're most likely to respond to?
And it's clear that we all sit and think about this from a drug development perspective and contributing good in that way, and bringing drugs to the market, helping patients. And I think another thing, as we're sitting here thinking about data for social good, and curious, your thoughts on that. How do we look at it from a bigger perspective? Because it's obviously a big academic consortium and has some interesting elements about that as well, and providing this to academics. I don't know, Paul, if you thought about that or have any.
Yeah, actually, I think there's a couple of things that spring to my mind on that topic. It first really touches upon what you just mentioned. There is this very brief period of exclusivity that we have with the data, but I mean that's really a blink of an eye in the grand scheme of things, and this will all be released into the public domain. And so many researchers around the world are already accessing and working on the UK Biobank data, and so this will be a really rich additional source of data that will be out there in the public domain. And you can think about the exciting discoveries that will come from that. And I think the other thing that I would add to that comment is there's many ways that you can look at the data, and there's a lot of very interesting academic questions that you can ask. If you have 500,000 genome sequences of individuals, there's many interesting things you can do. But what I'm really sort of proud of in terms of our involvement with the consortium is that we're hyper-focused on using that data to discover new medicines and to help patients. So in terms of sort of the broader social good, I think, this group of companies really being the genesis for that data and really being invested in using it, I think is also another, really beneficial thing that will come out of this.
Mm. I mean, it's an investment, collectively it's over 100 million. So, as it came together, I think probably a bunch of people paused on that point.
Yeah, absolutely. And I think part of the things is you can see we're all really aligned on how genetics and precision medicine will affect patient care moving forward. And not having one company be able to do that on our own, I think you can see we're all fairly aligned on the future and where medicine is going and what our contribution can be to that. So I think it all got us very excited to say we can pitch in a little bit together and leverage each of the resources we've all built. I think the added cherry on top is the fact that this will all be available to public researchers through an application process where they can go in and use this data for their own research and maybe at some point have a potential impact on patient care, although that would be something an academic and medical center would be more focused on. It has that utility as well, and I think that's just a huge benefit.
Yeah, and we were just not seeing, the biobank wasn't seeing a source for public funding for this. So I think that was the really exciting thing from their perspective. Without this investment by this team, we've never had this as a resource for everyone, and that's really exciting. I think that's I think it's really interesting to look back. I personally have been touching human genetics since the late 90s, early 2000s, when it was really like a complete pipe dream. And if you guys look back to the sort of last 5 years or so, what have you seen? I don't know, Joe, as changes, because I think it's really gone from a hypothetical to now something in front of us that we can touch and we can see. What are you seeing as the applications and the receptivity?
Absolutely. So I think a lot of the growth and uptake and acceptance of the role that human genetics has to play in drug discovery and eventually in the practice of medicine has been driven by the technologies that we're going to be employing as part of this consortia that will allow us to sequence 500,000 genomes in the course of about 2 years, right? So even 5 or 6 years ago, that was unimaginable. But to put this in very concrete terms, I think as a field, we've recognized that drug targets that have human genetics evidence behind them have turned out to be more likely to succeed and make their way through clinical trials and ultimately registration and onto the market. So as a recent example of this, just this week, BMS, we announced the results of a trial in arthritis where we have an investigational drug that targets the product of a gene called TYK2. Okay? So there's exquisite human genetics evidence that supports the role of TYK2 in a number of autoimmune diseases, including rheumatoid arthritis and lupus. And so historically, those types of analyses or results largely have come out of the academic community. So when we were presented with the opportunity to join this consortium, we realized we would be in a position where we could systematically do these types of analyses at a scale that was unimaginable even a few years ago, right? And really focus our attention on the types of diseases that we have expertise at in BMS.
I don't know, Salve and Morten, how have you seen the field evolve in your times in this area?
Yeah, so firstly, I agree with everything that was just said, but I can think of 3 other things that have really changed and allowed for this to become a possibility. I think the first one is the participant interest and engagement. I think especially in the last 5 or so years, the community has accepted contributing their data, clinical and genetic, to medical research much more, which is excellent for everybody. I think the people that benefit are the community. Secondly, as was just mentioned, within our companies, we are viewing genomics as very important. And if you look at AstraZeneca, in our clinical pipeline, over 90% of our current clinical pipeline has a precision medicine approach. And actually we have 4 FDA-approved medicines that come with a companion diagnostic. So what this means is you have a genetic marker that allows you to identify which patients would benefit most from that specific medicine. And the third part, I guess, myself just recently moving from academia to industry, so exposure to academia is fresh and exposure to industry is fresh. One of the things that's really been pivotal in my mind is this kind of paradigm shift from the traditional silo approach. This is my data as a researcher or as a group, and I don't want to share this because I think this is the best data out there, to a kind of the entire genomics community working together to break down these silos to allow for a much more fluid landscape of data sharing, because at the end of the day we all have the same end goal, to improve patient outcomes, improve patient care, to identify new drug targets and to create innovative medicines for the unmet patient needs. So I think those three things have really transformed data access.
I would point to, of course, there's been a lot of technological progress, certainly on the sequencing and DNA characterization, epigenetics, but also functional measures. A lot of high throughput methodologies for identify patients, proteomics and metabolomics and other measures. And because the genetics in itself, of course, is not useful if you cannot relate it to some trait or some measurement. So the fact that you can do that in high throughput really, I think, is kind of revolutionizing, what we can do with our data, and also where I see it, genetics moving a little bit more into the mainstream. I would say in a company like ours, that's increasing being realized. I think the other maybe biotech companies are a little bit, Pfizer tends to maybe, come, I wouldn't say we come later, but of course, any investment you make, it's always an opportunity cost to it, right? Because you could say, let's do whole genome sequencing of everything, but then there might be some programs that you couldn't run, So how do you, so you have to really be smart about what you're doing and how you're applying it, right? And I would say another, maybe last point I would make there that several colleagues made as well was, that really in terms of what we're interested in and looking for in terms of understanding disease, I would say we're fully aligned with, what typically the academic community is looking for, right? I don't know if it's always been like that, but I think that's also why these kind of pre-competitive consortia are very, very attractive. And actually, most of what we are doing in genetics really is working with academic researchers or peers in other companies. Of course, when it comes to our clinical trials, we have to be a little bit more cautious, as does the colleagues as well, right? But it's really quite an open environment, which I think is very inspiring.
It's such an incredibly rich resource for it. There's just really no way any one entity can tap into that. I think that's the other piece of it too. When you look at the size of this data set,
We're going to be busy for a while.
Yeah. And then I think the other piece too, you're talking about the technology, which makes me laugh because I think back to, the size of sequencing, setups and ones that I'm familiar with. We blew out a transformer once for the city of Cambridge. And now we go look at the sequencing efforts and you waiting to see how big this is, and these small boxes are just running 24/7, but still it's so different. But also the medical records, which I think have changed enormously.
Yeah, I think the UK Biobank made this, just great effort to go. And each of these volunteers have come and donated just not their sample, but their time and putting all their medical history into a single place, and that just makes a massive difference when you're looking at the genetic data itself and being able to parse out.
Right. But then that raises another point, which has been really important, is privacy.
Yeah.
So I don't know if you want to talk a little bit about how we've thought about that as a company and setting this up, and the importance to everybody here.
Yeah, so certainly, these are volunteer patients, and we get de-identified health records from each of these patients. All of that data is kept very securely in Amazon's cloud. Each of the companies here, have access to that. We take it very, very. It's a very stringent thing for us. We also have 70 other collaborators here at Regeneron that we work with, in addition to the UK Biobank, that all put their data and give their data to us so we can use that as a potential resource as well. So knowing in today's environment how important that is, that's something we take really seriously.
Mm-hmm. Yeah. And I think a big commitment all around by all the companies. I think it's something we're used to from working with clinical data, but I think now that we're looking at these resources that are so significant, that just really taking a careful look at all of that for for all of us. I think that's been really important. So I guess I'm curious what each of you think is going to be the first discoveries out of this. If you think of, there's so much we're going to touch and so much that it's going to bring to us, but what do you think are the near-term hits that we're going to each hit, near-term benefits?
Well, I think, coming back to the points that I made at the beginning, there are these really like two major groups of questions that we want to ask. One being, if we can identify individuals that carry mutations in certain genes, and we want to look at their medical records and know how they're affected by that mutation. And then the other aspect is trying to discover something completely new. So studying a disease and trying to find something novel. So from our perspective at Alnylam, I mentioned at the beginning, we have our first approved drug. Actually, we're the earliest stage company of the group here. So about a month ago, we had our first drug approved, which is for a genetic disease. And we have two other drugs in the pipeline, which are relatively close behind, also targeting genetic disease. And what's really interesting about all three of these diseases that we're going after is that they're actually incredibly difficult to diagnose because they present in a very general way and they could be mistaken for other things. But we know, based on the frequency of individuals that carry these mutations, that we'll find a fairly significant number of them in the UK Biobank cohort. So what I think is kind of our low-hanging fruit is simply to ask the question, well, if we find these people in the data, let's look at their phenotype. Let's look in their medical history and records and understand what are they diagnosed with, how do they present, do they always have the disease, do they not have the disease? If they don't, what are the other factors that can influence whether the disease manifests or not? And so that really will feed into our efforts to help to bring our medicine to as many patients as we can. We want to be able to help folks with the drug that we've had approved and the other molecules that we're developing, but we have to do a better job of finding them. So I think that's going to be, for us, one of the first things we hope to get out of this.
I don't know if you have thoughts.
Just a comment on sort of the nature of the data and going back to the technology. So if we can kind of geek out for a second, right? Because this is such a large data set, 500,000 subjects, it means we're going to have an opportunity to find really rare private genetic variants that you wouldn't be able to find in data sets that were a smaller size. And because of the technology that we're using, doing actual sequencing, rather than just looking and measuring a handful of landmarks across the genome, we're able to find the actual variants that cause the phenotypes that we're interested in. So what that means is we have this opportunity. Regeneron, you guys have a fantastic example of this past spring, and the NASH target that you described in the New England Journal of Medicine, to identify really causal genetic variants that give us very strong hypotheses and evidence about the roles that specific genes might play in clinical or sort of phenotypic signals of interest. That's huge. This, just a couple of years ago, this would not have been doable at this type of scale.
Yeah, I mean, just to add to that, I don't know if you're aware of that, but the general way, how we would usually do is genetics with this SNP genotyping, where you rely basically on 1 out of 1,000 nucleotides that there's a variant, and that there's a need to find a functional consequence that inquire whether there is a functional consequence. But that works for some genes where you see a strong signal, but actually for many of the genes that we could potentially be interesting, there's no such signal. And I think it's alluded to, as what we can do with this data is, we basically in every gene we will have these rare variants where we can, the hope is that we will see many examples where we have a strong effect on disease. Something else that we are quite excited about is when you have such a large data set, we can look for what we call resilience, you know, mutations where you have, for example, you know, a gene or variant that you know is detrimental, or let's say like ApoE4 for Alzheimer's, for example. Then you can ask the question, can we actually find genes or variants that would protect individuals who have that variant from actually getting the disease? And if you can find such you know, such insights, I mean, that tends to be very strong drug target, right? So that's something we're excited about. We can also use it for what we call structure-function relationships. You know, for example, for gene therapy, you can go in and see, you know, in specific genes where you want to make a gene therapy, you know, what are actually the mutations you find in there? Do you even find some mutations and then actually it turns out that the individual actually does not have the disease? And that will tell us something about, you know, how we should both design our gene therapy and also maybe do patient selection. So that's something we're quite interested in as well.
Yeah, I'm just going to add to the previous comments. Really, I think a key advantage of this data set over the existing data sets, including the UK Biobank common variant data, is exactly the access to the specific individual variants that are potentially unique to those participants. And the importance of this is really great, because if you think about it from a kind of the contextual point of view, there are many ways, for example, you can break a gene. And so there isn't necessarily an expectation that, you know, one way to break the gene is going to be the one that's present in all these patients. So if you think of, you know, what we call loss of function variants or protein truncating variants, we've been blinded to a lot of these in previous genetic research on the basis of using the earlier technologies, whereas now with the sequencing technology, we can take the 500,000 individuals and we can get a larger population of individuals who have different mutations in a gene that are probably private to them and their families, but those mutations have similar effects clinically. And so we can start to have more power to identify clinical correlations with a class of mutation rather than a specific mutation. And that's, I think, the really game-changing part of this technology.
If I add one more thing here, I think the UK Biobank resource in itself, I mean, you guys have seen this too, many of these cohorts that are built are either built around one certain disease or one phenotype. And the UK Biobank is just this resource that has, you know, dozens of phenotypes and dozens of diseases that are being collected. And there hasn't been a resource that's been generated that where we can, essentially one resource that we can look across all of these different phenotypes. And each of the companies here, yeah, we might have different therapeutic area focuses and business interests, but this data set, you know, we could be making available dozens of diseases where academic researchers can go in and study what might be actually causing Alzheimer's, or some of these really high unmet medical needs that just, you know, we haven't been able to crack. I think that's what's really exciting about it.
Yeah, no, I was just thinking about that. No, from an academic perspective, how are they going to be using it?
Exactly.
You know, because there's just hundreds of ways that they'll tap into it that, you know, probably we won't be able to get to it, or it's an approach that is just not, you know, we're not looking at from that perspective, or we can't actually develop a drug in that particular space, unfortunately. So it'll be, I think that'll be really exciting to see what comes out of it from that perspective.
Yeah, you know, just struck me was that, you know, I think actually an important role we have here is, you know, I think actually UK Biobank has done a really good job of putting together this huge resource. I think academic researchers sometimes would be a little bit, you know, this is my data, you know, and so we actually, I think, by having such an initiative, we actually drive, you know, kind of building a large resource that, you know, everyone can use, you know. So I think actually we play quite an important role in the kind of in the ecosystem. And
No, I was thinking about that before. Like, do you think this will, you know, have you more inclined to other pre-competitive? Because I know, you know, probably when you went back to your organizations and said, like, you know, I'm interested in this investment, and by the way, it's going to be publicly available, there was probably hesitation. Now, how does that, you know, how does that make folks feel about it? I don't know.
yeah, that's good.
Yeah. What was your experience?
It's a little, it's a little bit paradoxical.
Right.
If you don't join in, you know, the likelihood of the data being generated is, you know, goes down. So I think what we have to do, and, you know, I think this point, and, you know, one thing to really recognize here, just to echo, is that there isn't anything like this. Right? This is like, you know, in the history of mankind, this is the first time this type of data will be kind of broadly available. There have been other large sequencing projects dotted around the world, but as you've heard, those have been kind of kept closed. So this is open. It makes it very unique. But I think what we have to get away from is sort of thinking about generation of the data as some kind of competitive advantage. I think that's just nonsense. I think the advantage is all the creative ways you can think of mining the data, the questions that you can ask, and there's an abundance of those. People will have different ways of doing it, they'll have different questions that they can ask, and I think that's, you know, the sort of premium, and that's more to do with the people that you have on your team and the things that your company is interested in, etc. So I think building and sort of, you know, expanding these data is really just going to benefit everybody. And so I think that's a really kind of key thing that we have to bear in mind.
So just to add on that, I think, you know, thinking about data stewardship, data governance, data sharing, one of the things that I expect all the companies had similar experiences was that, you know, you set up to go into partnerships with the shared vision and a shared roadmap, right? That you're aligned on a roadmap, and the end goal for all of us is improving patient health, patient care, patient outcomes. Again, you know, identifying new targets, genetic targets, developing, you know, new innovative ways to address unmet clinical needs and so forth. And so when you kind of have that shared goal, all these discussions about data stewardship, data governance, they're still there, but they're so much more streamlined, because at the end of the day, you know, you want to do the right thing and you want to make it available, because in my mind, the more people working on it, the better the outcome will be for the population.
But also, I mean, I think one reason that it works well to do pre-competitive collaborations here is that often, you know, where we have, you know, IP, you know, what's the most challenging, that's around our products, you know. This is an area of, like, typically open science where, you know, you publish the results, you find some new findings, and it's nothing that, well, at least it doesn't translate into kind of IP around products, you know, so straightforward, you know. So you know, I think also makes probably many companies open and more comfortable collaborating in that space. And to your question, it's good to have like a few months exclusivity where we can play with the data. You know, it's like $several hundred million, you know, so particularly our senior leaders, right? I mean, but you know, then of course there's also that obligation. I mean, the UK Biobank, the UK government put in hundreds of millions, I don't know if not billions. I mean, they put in a lot of money in that. And of course, there's an obligation, you know, that we that we are good partners and citizens in that context as well. So.
I think one thing that we've seen, I mean, not just through this effort, but also at Regeneron Genetics Center, maybe you could speak to this, is the difference in patient involvement, which is fascinating to see. I think that's been really interesting. I don't know if you have some comments on
certainly not
explain
even just
to
from
me.
our perspective, but I mean, all of you, I'm sure, you know, 23andMe and Ancestry.com. I mean, there's just this heightened awareness of how genetics can actually impact not just, you know, what genealogy of where you came from is, but also, you know, do I have certain things like a BRCA mutation, and how can I, you know, if I know that now, can I make, you know, some sort of decisions in medical care, you know, to prevent certain diseases, or can I change my course of care? There's just this heightened awareness, I think, just industry, you know, within society overall, that we've seen. And at the Genetics Center, you certainly, you know, we've seen this heightened awareness of our partners in these academic medical centers and health systems to want to include, you know, genetics in their everyday care back to patients. So just having everyone just elevating, you know, the commitment to genetics throughout society, I think is hugely valuable. One of you guys made the point of, I think it was you, Salve, that said, we just have this increased patient participant, and people are generating donating samples and donating their health records, and that's something that 10 years ago we didn't have. So the increased awareness of people, you know, willing to use their data for research, and I think also our increased awareness of saying we want to generate this data for the public good is really something that's really important for everybody here.
Yeah, I'm based in Cambridge, UK, and I think weekly I come across somebody who will mention, oh, I'm part of the UK Biobank, oh, my parents are part of the UK Biobank. So it is it's a very big initiative.
Yeah.
I think in general we maybe tend to underestimate the willingness and enthusiasm for patients or citizens to participate in this type of research. This is internally when we have debates around what we're going to do in a clinical trial and what types of samples do we collect. Oftentimes we get pushback from our operations people about, well, if you include this additional collection, nobody's going to enroll in the trial. That generally doesn't happen, right? Because I think for the most part, people who have serious diseases want to contribute, right? They want their experience and their data and samples that they can provide to help others, right, overcome these diseases that they're afflicted with.
I mean, we've even had experiences now, we know of some hospital institutions where they ask at admissions, right? They sort of say, are you willing to give a sample? And just, that's part of your check-in procedure.
Yeah,
and
all
it's,
these
yeah,
consenters,
yeah, that just come in, and which is pretty amazing.
Yeah,
and then
it's
people who proactively, look to us, and there are already some interesting stories that have come out of, that where, people have looked up, we know there's instances where there have been ER cases where people have thought it was just been, a passing disease, and they sort of, okay, we have that data, and like, whoa, it could be something more serious where you wouldn't have even thought to look at that.
Yeah,
so,
exactly.
which is pretty amazing and pretty, satisfying
Yeah,
too.
definitely. I mean, there's just outside of just discovering new drugs. I mean, to your point of this is we're doing this to improve patient care. So you see our partners wanting to do this kind of, the first point of contact with a patient. I think that's just going to elevate genetics just overall.
I mean, I would say we don't have the same experience, right? That in general, patients are very willing, and to, give samples and share the data. Where we're sometimes, of course, we have to be very careful, with the privacy and integrity around that data. And I think, probably pharma companies in general are the ones who have the most to lose by this breaking down. In addition to the patients, of course, I think we're all, paranoid about really being, making sure that is done well. And something that we're struggling with sometimes is, what data do you give back to patients? Because of course you can give them all the data back, but they don't always know. Even us as some experts, don't always know what to do, with the data when we see it. So we have to be careful. I'm not giving the patient that maybe makes them uncomfortable and make their life miserable, or so. Or if there is a serious mutation and there's no cure, right? I mean, how do you handle that, right? So it's important really to think carefully about how we do that. But I think it's a journey where we continuously know more and more about the individual variants and more medicines becoming available. So it's exciting.
Yeah, I agree. I mean, those are really important points. And I think if we look at not too far in the future, I do honestly believe that we'll be in a position where it'll be atypical to come across someone in the street who hasn't had their sequence data generated. Right. It's just, it's going to happen in primary care, at birth, etc. And the real challenge and I guess opportunity as well that arises is how do we improve our interpretation? The informatics obviously is always going to be a challenge at that scale. But the analytics, how do we mine that data, to Paul's points earlier about getting, identifying those patterns that allow us to determine and to advise appropriately our patient populations? I think that's where a lot of the focus is going to be in the coming years.
No, it's an important point, and like you said, as we get more of it and it becomes more pervasive into how we, in medical care in all ways, then, thinking about how we get that data analyzed both, within our world, but then also sharing that back with the patients, and and patients are being proactive and approaching us about how can I contribute, and what is it that we want to make sure that we can provide back, not just in the discoveries we make and the drugs hopefully we can develop and provide, but also thinking about what we can do for the for the greater good for the medical community and for those patients. So maybe we'll leave it at that. Doing okay on time. Great. We'll wrap with that. Thank you. Great. Thank you.